the inside. So that looks like a sponge on the way out. Would you agree, Erica?
Correct.
That's good, thank you.
Sure thing.
So just to reorient people as we scoot along here, we're off the area of Morro Bay in a place called Santa Lucia Bank.
That's a black coral there. Center screen.
All right, Marley, go ahead and push on in again, please.
Is there any interest in sampling that?
Liz?
Is that a chrysopathes?
Don't know.
Very pretty, aren't they?
Yeah, I would tend to say lilipathes, maybe.
Oh, yeah, you're probably right. Just the, yep.
Great. All right, Marley, come wide, please.
Do we need any lilipathes samples?
I've got so many tabs open. Let me check.
Okay. I know we're always trying to unravel the bathopathes, lilipathes, chrysopathes issue.
Yeah, let us know if you want the
I think
sample.
we can.
Yeah, I can stop the show.
Because I want to stop the show.
I think we can just keep moving.
No, I think keep
Go ahead.
going.
Roger.
Roger that.
We got some good great images of it. So
Trying to think what the Latin root pathes means.
it is on our list, Liz. So if we see another one, maybe grab a snip.
Yeah. You see
The
another
flytrap anemone.
I'm sorry, what did you call that anemone, Erica?
A flytrap.
They do sort of look like a Venus flytrap when you get up close to them.
And it functions like that. Something gets inside.
I don't think so.
Okay.
But
Like has what go on.
I think it's more based on their morphology, the way they fold like that. That's a pretty common form.
So as I was saying before we stopped to look at that black coral, that we're off of Morro Bay area off the coast of California. We are in an area offshore in deep water in an area called Santa Lucia Bank area. And we've been diving since about 6 p.m. last night in this cobbly area that seems to be populated with scattered sponges and corals. The primary fish has been a grenadiers down in here with some skates. And I think we saw a deep sea sole.
Lots of the pineapple benthic siphonophores.
Yeah, those things are fascinating. They attach themselves to the bottom with one of the tentacles and float above the bottom, but attached, unlike a lot of other siphonophores, which are free floating. These guys are like little dandelions with the stem. What's that we're coming up on, Erica? Oh, an anemone. Never mind.
Marley, push in, please. Oh, wonky shaped anemone.
Pretty
Isn't
though.
it?
Yeah.
Yeah, it's having a bad hair day.
It's a punk rocker.
You use too much hair gel.
All right, go ahead and come wide, Marley, please.
And those white, long, snaky things are sea cucumbers.
And so is the one going off to the bottom. That's a solus, more of a compact
Yep.
armored sea cucumber with its feeding
Yeah, that's the
tentacles
first.
out.
I think that's the first solus I've seen on this guy.
So we should be getting a little steeper during this last 100 or so meters.
Roger.
More dead sponge off to the left there.
Looks like it.
Then a live sponge coming up in the
Yeah.
distance there. You see those sponges where they provide habitat for a lot of things.
All right, Marley, click zoom there, please. That's great.
Those are very cool crab.
Yeah, that's awesome.
Wow, that's a good view.
Spiky. Spiny.
And ready for my
Spiky.
close-up, Mr. DeVille. Very spiky.
Thank you.
Look at that guy. Now that's some hair gel.
Yeah, that dude is fierce.
Its mouth parts are right in our face.
Yeah. All right, you gotta move. All right, go ahead and come wide there, Marley. That's a cool little community.
I'm paging through guides here, but I can't put a name on that guy.
I would guess that's another one of those neolithotid air quote king crab family.
Do they get larger and that's just younger?
Those were pretty young because the spines were still quite defined, and the older ones, they do wear down. But we just don't get the size crabs that they do up in the Bering, the king crabs.
Definitely look like those spikes were fresh.
Fresh spikes, exactly.
Fresh spikes.
The sea cucumbers sure do like this cobble.
Yeah. Can we zoom in on that? I can't see what it is, whether it's an anemone or a sponge. It's about in the center of the field of view right now.
Roger that. Stand by, Marley.
And another anemone.
Very nice symmetry.
Yeah, that one's having a better hair day.
Yeah. All right, Marley, go ahead.
Thank you.
Yeah, and just so you guys are aware of time, it's going to take us about an hour and 15 to ascend, plus 15 minutes on the surface. So we'll probably want to get off bottom around
Combine, please.
6:30. And we're getting pretty close to that that target to start our ascent. And
Okay.
yeah, should take us,
I
so that ascent will be about 23 minutes. So we should just get it. One thing I noticed in the slope map is it does look like there's a little steeper slope to the south of us, but it's also a steep part right on that upper part of the ridge. So we could try to do a little zigzag, slight zigzag, which shouldn't add much. This.
that makes sense to me if we have time.
Okay.
Okay. If we stop zooming on things, if we stop making them stop, they'll do better.
I wasn't gonna say it. No.
Yeah.
No.
I
Making good time. And also, it's like the ship moves, her can catch up, so the zooms are
Yeah.
much of a problem.
Sometimes we just zoom to not stare at empty rocks.
Dawdling scientists. We could have an ROV dive where you actually don't go pretty much anywhere. Just take a look at everything around in one spot.
I love the concept of just zooming, and you just see very different things every zoom.
Right. The cameras you guys have are so amazing.
You must get pretty accustomed to your perspective from the AUV habitat mapping.
Is this the same coral that we passed by earlier that was on the collections list?
It is.
Shall we grab it, Erica?
Sure. Let's grab a snip if we can do it quick. I know Lizzie
Bridge
and
nav,
Lisa
can you hold position?
are interested in that slope.
Roger.
It looks like a pretty easy, just cut and go. Well, easy for me to say, sitting here with my cup of coffee in my dining room.
Looks like we got a topside crash there. There's little widgets hidden under all this Grafana stuff.
Bridge nav, can I get 20 meters 315?
Oh, that doesn't work anymore.
So I think when they say a topside crash, some of the software is acted up and they have to
Thanks for
restart
backing
it.
it up. Thank you. Yeah, just enabling all the things again.
The common language between ROVs and AUVs. Topside is the software on the ship, and sometimes it acts up.
That was a 20-meter move backing it up there, Ben?
Yeah.
Great.
All right.
That works.
We'll probably just go directly up the slope after this.
Roger.
So we think this is a Lillipathes.
That
Okay,
is
Marley, go ahead and
my
zoom in,
first
please.
guess.
Say again, Erica?
That would be my first guess, but
Yeah.
I have been wrong before.
Well, a lot of people are wrong. These on these. That's why we're collecting this one. Oh, gorgeous, gorgeous. Is
All right, come away, please.
because we're trying to. There's a bunch of black corals that are very hard to tell apart, and partly it is because we've really never had enough samples to closely examine them.
All right, and all right, I think I'll go for one of the smaller starboard bile boxes, unless you guys wanted this to be slurped.
If
No,
you
I think
can
it's
do a
not a
bio box,
slurp.
that's great.
Great.
Yeah.
All right, Marley, go ahead and flush in a little bit.
Swimming crinoid.
How much of a haircut do you guys want with this?
Whatever you can grab. One or two of those branches is great.
Sure thing. Ooh, that's a lovely view.
It may be pretty tough though. Not cut very well. Oh, you got it. Great.
Yep, looks good. Picking a bouquet.
Yeah. All right, you guys got your frame grabs there? Mary, you all set?
Yep, I'm all set.
All right, full wide there, Marley.
That looks great.
Mary, remind me of the starboard bio smalls that we have open.
We have C and D open.
C and D, Raj.
Want sample?
Oh, yeah, sure. All right, Kim. Thank you.
Lizzie's saying they collected a similar black coral on the last shift, so this will be good. We'll have two to compare.
Let's try that again. most elegant of samples. Come on.
Oops.
Neat. Okay, let's try this again.
So we're off Santa Lucia Bank, California, off the Morro Bay area, and we're just collecting black coral here. We're in an area with lots of cobble, with scattered corals and sponges, and we're at about 1,200 meters of water depth.
The fact that it's branching. Let's just try letting go, maybe, perhaps.
Pretty floaty.
Yeah, pretty floaty, that. Well, that's a floaty coral.
Ooh. Bye.
All right, we'll get a small snip and just shove it into the box.
So they grabbed one of these before?
I think these have another black coral that they grabbed before. Looks quite big. That rock behind it. I'll just get a small snip. Riley, you want to push in there,
A
please?
small snip is fine.
Roger that. Yeah, we might have to kill the verts. One of the isolations on one of the verts there. I'll just have to reinitialize it pretty quickly, I think. Smaller bits, but this will stay. All right, go ahead and come wide there, Mike. Thanks, Kim. I'll get my arm down in here, and then we can kill one of the verts, and then reinitialize it once I get my hand out. All right, opening now. Go ahead, Kim. And roger that. Anyone press stick lock, please?
Yeah.
Thank you. All right, go ahead and close up that box, Kim. Oh,
Nice move.
awesome. Thanks, Kim. Thanks, Ben.
Nicely done.
Oh, Raj. Thank you.
So science, we have about 30 minutes left, 35. 40.
Okay, I am going to stop talking
No.
and just let you guys cruise.
So do you want to start heading up slope now? And we can just go due east and try to hit some steeper features.
Yes.
If you
Sounds
look,
great.
okay, all right.
Roger that, due east.
Yep.
Thanks, Kim. All right. Sounds good.
All right, I'll wait for
All right, Ben, go ahead and call it in.
Looks like some more of that treated dictum sponge down there. We grabbed a sample of that. We need to come up with a fun name for these spiny crabs, the spiky crabs. Snailfish cruising by.
Yeah, I would like that, but that is like 160 meters away. So I don't know if you bias it, it's not going to give us much. You could do a little zigzag. I'm just worried about time.
Oh, nice. Science, there's like 2 options we could do right now. Thank you. Ben, you want one?
Oh, good. Yeah, we could do a little zigzag to the south and then back up. If you're looking at high pack now, there's a slightly more of a slope to the south. It's like 130 meters. We could go in that direction, and then about 130 meters back northerly. That provides better coverage and perhaps steeper slopes.
Yeah, or else we can continue due east and then just kind of go immediately upslope.
Yeah, because we're about 150 meters away if we go due east from the steepest color.
Erica, what do you think? I'm leaning toward just headed to the slope. What do you think?
That sounds great.
Okay, let's do that.
All right, due east.
We're on our
George.
way. Yep.
Thanks, Ben.
Another deep sea sole.
Erica, is there any fishery for deep sea sole? I don't think there is there?
There is not. They have a high water content like the deep water Dover.
Oh, so they're kind of mushy.
Very mushy.
So Dover sole that are in deeper waters tend to be really mushy, and the ones in shallower waters tend to be more, I guess, normal, kind of fleshy, and those are the ones that the commercial fishermen target. If they go too deep and they get these mushy ones, they're really not desirable at all as far as eaten fish go.
Looking at your heading now that we don't have Grafana. We don't have that. It's hard to see. Yeah.
We just took a sample of a black coral a little while ago, and the reason we do that is that they're hard to identify unless you have them in hand. And also we can do genetic analyses on them that Meredith Everett will be conducting. We should remember to take a bottle at the end just before we surface, okay?
Roger.
For Meredith.
Just
And
before
those
we
water
ascend,
samples
or?
that Yeah,
If we get a
before we said-
transect in.
So the only real time we have would be to do it now, and it would be going upslope. That if there's a
Then
transect.
let's not do it.
The bottle or the transect, Liz?
Did you mean the bottle or the transect now?
I just was saying that for the transect, we're going upslope right now.
Shall we start a transect then?
Yeah, we're running out of time.
Yeah.
Yeah, we can do a transect. We just won't be
Yeah,
able
it
to
should
stop
take-
and zoom and things, and
Exactly.
It
should take us to almost exactly the time we want to recover.
yeah, so it'd be pretty much the
Perfect.
last thing we do.
Okay, so
Sounds good.
start one now and then pull a bottle at the end.
Sure, we can do that.
Roger. All right, I'm going to mark it now.
Get a
Position, and Jess can get her autos going.
It's one of those Venus flytrap or flytrap anemones.
Mary, this is transect 7.
all right. Yeah, sorry.
I have it as 7, yes.
Okay, Raj.
Alrighty. Ready, Ben?
Yep.
Stepping forward. And science is the start of our transect.
Copy that. Thank you.
Sure thing.
Bridge.
Oh, we don't have our
So when
lasers
we
on.
say we're doing a when we say we're doing a transect, it means we're doing a line at a sort of in a specified way. So then people can do really a quantitative analysis of the abundance and distribution of the biota here.
Hey, Cam, you want to come up on the delta, please?
And so that's what we're doing now. We have a specified way to do those, with a general height, speed, and then it allows us to compare different areas, but sampled in the same way.
Thank you.
Those bathypathes are enormous. I wonder if dyes count the siphonophores. Another bamboo. Ooh, that's a different coral.
What's your guess on what that was?
Kim, Kim, you want to come up on the delta, please?
Lots of dead Pharea.
It's a good shadow coming up there in sonar.
Yeah, I see. I think it's
That's good enough.
been staying around the same distance.
Heteropolypus mushroom coral.
That's the first one I've seen of those on this dive.
Good morning, Abby and Meredith. They've just joined the science chat. That looks like a Hipposteria sea star.
Abby and Meredith, we're doing a transect right now.
Another mushroom coral.
But we've gotten on this watch and the previous watch some black coral samples. They should be interesting, Meredith. Ooh, Meredith is saying she's excited for the black coral. She just looked at, reviewed some of the earlier videos. And Abby, we got sponge. What does Abby think that sponge is that we collected, Erica?
A dictum. So this coral
Sorry, you
coming
broke up there.
now, Meredith. Meredith, can you see the coral coming up right now? Any ideas on that one? Yes, the red.
She's saying
Some
it's
flavor
a sword of some sort.
Different
Meredith,
kind
we're
of plate
planning on
sponge there.
Meredith, we're planning on taking a bottle at the end of this transect. Meredith is doing an analysis of the water bottle samples for environmental DNA. That's DNA that she can, that is in just the water that is shed by the local biota. And she can analyze the water samples, look at what DNA is in there, and use that as a way to tell us what things are in the region.
Mary, are we taking a sample at 200 meters and 50 meters again? Depth?
If we have extra Niskins, yeah, we can.
Roger.
I wasn't told specifically that we needed them.
Okay, roger.
Yeah, we've come about 120 so far on this transect, and it's about 617. We were thinking of coming up around 25, 620, 625.
Sorry, for the come up time?
Yeah, I'm just trying to make sure.
Yes, I believe it was 6:30 around we can come up.
Let's see. Yeah, so our ascent should be just about an hour and 10, hour and 15, or sorry, hour and 20, and then plus 15 minutes. So
Yeah. I'm gonna drop a weight while we're on our way up so we can go up a bit faster. Sorry, I'll hold off here.
is that a team? Oh, yeah. Low 18. Strange color. Like fragile looking.
Seeing some crinoids there in the field of view. Those things that look like little feather dusters.
Liz, do you see the ctenophore right in the center?
I do.
Yellow and red. Yeah. Really see the stone medium with the pharynx. That's the red part.
there's a ctenophore. I think it's called a blood ctenophore.
Oh,
And that may be what this is.
sounds like an appropriate name.
There's an eel pout, probably a 2-line eel pout, heading out on the bottom left.
There's something called a blood belly ctenophore. That may have been what we just saw.
We're racing the clock on our final transect of this dive on Santa Lucia Bank.
It's that blood belly ctenophore looks like it's a lobate ctenophore.
I think that
Ben, what's the distance we've come?
yeah, so we've come about 130 meters. So
Roger.
So for those of you who are just tuning in, we're just finishing up this dive. It started